This is Chapter 1 of Microbiology. I'll be working mainly from the Tortura textbook. That is the textbook that we'll be using. And this is going to cover introduction, history, and nomenclature, which is the science of naming. All right, so we're going to see how microbiology got started as a subject. And I first wanted to start by introducing the topic with just a little bit about what makes microbiology interesting, like, what about the microorganisms in our lives, the microbes. And so in Chapter 1, it starts out with a chapter opening question about advertisements. Advertisements that are all over the grocery stores and anywhere in which you can buy antibacterial products, cleaning products, hand soaps. And the question poses whether or not these products are good, are they preferred, or in fact, maybe are they not preferred. And you've probably heard evidence to both sides. So that would be one of the questions that you should ask yourselves. And so I've posed this question before to my classes, and I've got a variety of answers. And since we're doing this remotely, I'll sort of talk through some of the answers to this question. First of all, a lot of people just jump to the idea that no, maybe we shouldn't use specifically antibacterial cleaning products in our household. And so why would that be? I think that this would be one that we can discuss further in our Zoom meetings. So I'll just plant sort of an initial thought, which is that a lot of people have heard about the fact that not all bacteria are bad, and not all microorganisms are going to cause us harm, and in fact some of them are beneficial. And so using cleaning products that destroy all the bacteria in their path may not be necessarily a good thing in all cases. So for no, the angle would be that beneficial so-called normal flora may be eliminated. Okay, so something that we can get to in a Zoom meeting is we can discuss the answer to this question further. We can look at other reasons why we might not want to use specifically products that are labeled as antibacterial. But let's not go too far with the idea that sometimes we don't want to control bacteria, even in our own households. Think about times when you're in the kitchen and you're trying to figure out, hey, what about that raw chicken? Maybe I need to sanitize the surface. How about your toilet, right? You go and you use the bathroom, you're going to scrub it with maybe some soft scrub or something with chlorinated compounds or chlorine itself to destroy those bacteria that we really don't want, mucking up areas that we want to have squeaky clean. So the answer would be yes in some cases as well. It's okay to kill bacteria where you really don't want them. Disinfection is an important procedure. Disinfectants and antiseptics. Okay, disinfectants are used on inanimate objects, and antiseptics are used on living surfaces, like mouthwash for your mouth, or say a cut or a scrape or something like that on your body. So disinfectants and antiseptics will kill potential pathogens. And in that case, you really do need, by the way, pathogens are microorganisms that can cause disease. And so sometimes we do want to use antibacterial products specifically for that. So we'll get back to this question. There are other reasons why we could say yes as well, especially if you're going to be a healthcare professional, as most students are taking this course to very much try to control bacteria and other microorganisms. So again, we'll discuss this further in our Zoom conference. All right. I would also like to just define what microorganisms are. Microbes and microorganisms refers to the same thing. A microorganism is really any invisible life form. All right, so it's got to be something that is alive, although viruses can debatably be a life form in the sense that they are put in with microorganisms, even if they aren't able to reproduce as other living organisms that are complete life forms. We can also talk about germs in a more casual sense. When we say a germ gets on you or in you, it could be any number of different microorganisms. Germ sounds a little more casual. Our book defines it as any rapidly growing cell. We think of like a germ as something that could get in you or on you and start growing and multiplying so that it colonizes you over time. And so germ is an okay term as well. All of these terms we'll get used to using to refer to the various microorganisms. And we're going to define what all the different microorganisms are, and so we'll see that coming. Okay? We should keep in mind that very few microorganisms are actually disease-causing, known as pathogens. And so pathogens are what we're going to pay special attention to, since most of the students in this course are going to use it for healthcare, and you're here primarily to learn about how to control disease so that you can help treat your patients or be aware in a professional medical setting on how we'll take care of those pathogens when possible. But many microorganisms, the vast majority, are nature's recyclers, or they help us in positive ways to digest our food, to give us the oxygen that we breathe, along with the plants, of course, we know that give us oxygen. And also microorganisms produce a lot of other chemicals or useful items for us. And so you can look at some of the things that are in Chapter 1 that microorganisms can give us, ranging from explosive items to things that we use to make food. And so microorganisms can be really exciting to talk about when we start thinking about things like that we can consume that without microorganisms we wouldn't have, like yeast, for instance, right? Yeast makes amazing bread and pizza and pretty much anything bread-based, or even to ferment, so wine and beer, as well as vinegar and certain cheeses. Without yeasts and bacteria, mold even, then we would not have some of the amazing foods that I think a lot of us find to be quite, exciting. So besides that, there are also biotechnological advancements that without the bacteria, we would be really, without a lot of the tools that we rely on every day. So the biggest example biotechnologically that's been around for decades, half a century practically, is insulin. And still to this day, it's a vital chemical that people need to inject if they're diabetic. And so someone who has diabetes, they're able to receive that insulin from E. coli, surprisingly. So the same friendly E. coli that generally will live inside of at least all human gut tissue to help us to digest our food, those E. coli can be genetically programmed to make human insulin. And that wasn't a small feat. Before that, diabetics would have to get insulin from cows, bovine insulin, or pigs, or horses, or something like that. And in fact, those sources of animal-based insulin are not as good because they're not exactly like human insulin, and there can also be sensitivities where someone who's diabetic would then have difficulty with maybe allergic reactions to the animal proteins in the foreign insulin. So having insulin now that's produced by E. coli, which produces the actual human insulin, where we took biotechnologists took genes, the DNA for producing human insulin, and put them into the bacteria. And so we're going to see about that later in the course, how bacteria and other microorganisms can be reprogrammed to produce things that are desirable for us. And hopefully we're not programming those microorganisms to do, well, evil things. Bioterrorism is a real thing, and so that's another fascinating subject that relates to what we talk about during different parts of the semester. All right, so why do we want to know about microorganisms, right? Why are you taking this class? And the two main reasons that we study microbiology, the major reasons are to prevent disease occurrence, especially if you're a healthcare professional. So we'll focus on that. It's not a medical microbiology class, but we're going to get as much mileage out of that as we can, because I know that's what most of you are here for. And then you have preventing food spoilage, and everybody deals with that. Just putting your food into the refrigerator is a way of preventing food spoilage. And we use sometimes preservatives. Not all preservatives have to be bad. The word preservative could just simply mean putting more salt into food, and then the food is both preserved and tastier, depending on what it is. So we can look at preventing food spoilage in many ways, and we'll talk about how preservatives work to stop microorganisms from growing. And so we're going to focus, as I was saying, mostly on preventing disease occurrence. And one of the things that you guys are going to see are the laboratory techniques that we need to be aware of, so that we can practice what's called aseptic technique. Aseptic technique is the idea that we need to keep all of our professional instruments, whatever you're working with in the hospital, medical equipment, you need to keep it clean and free of contamination. And very importantly, we need to also make sure that we don't somehow transfer those microorganisms, that contamination, if it does get contaminated, into someone else. We know for sure that germs spread from one patient room to the next, from one patient to the next, and sometimes it happens through the handling of unwashed hands, right, where medical personnel don't wear gloves properly, or they're not sanitizing, using hand sanitizer or washing their hands when appropriate, and also the surgical equipment has to be sterilized or sanitized appropriately. So this is going to be a big one. This is the reason why probably 98% of my students are here, is for this. All right, so keep that in mind. These are the things that we're going to really be trying to, have some mastery of in this course. Now, we're going to go now into the naming and classifying of microorganisms. This is nomenclature. I'm going to write that down. Probably should have put that as a title on the slide. Nomenclature. And this is the science of naming. We have a clear set of rules. Well, at least we try to have a clear set of rules. And sometimes people argue over the science of naming and what those rules are, and they can change over time. And the people who are professionals at nomenclature are mainly taxonomists. All right, taxonomists are scientists. Some of them are microbiologists, and what they do is they make the rules and they make the names so that we understand how to classify microorganisms. And over time, we learn new things about the microorganisms, and we figured out that sometimes the naming system or the way in which the names are assigned have to change. So we'll see over time how that applies. But we have to first understand nomenclature's origins. The modern form of nomenclature that we use today was devised by Carolus Linnaeus. So in 1735, it seems like it is ancient history, but relative to maybe how long humans have been around and speaking language, this is a fairly young part of science, right about close to when microbiology started as well, as we're going to see. And that's kind of an interesting fact about nomenclature, because nomenclature you would think would predate the invention of the microscope, which is when microbiology started. But Carolus Linnaeus was born Carl von Linne. Is that said here somewhere? No, I don't think it is, but I'll write that in just because it's kind of interesting. Carl, how should we put this? Born Carl von Linne. And Carl von Linne was very much a biologist, someone who was looking at plants and animals and various life forms that he could see, and trying to find a way to give meaning to their names. And he used Latin. So it says here that his naming system was done by Latinizing the namings, and interestingly enough, he Latinized his name. So Carolus Linnaeus kind of sounds like a genus and species name. Kind of amusing. But Latin was a big language in Europe, and it still is today a huge influence over medical terminology and scientific names. So a lot of Greek and Latin comes through in what we learn about as you become a doctor or a nurse, or you're in the medical field. And I find it to be interesting. I wish I understood Greek and Latin, because I feel like I would just more naturally know what so many of the words that we come across mean. And here we see that what Carolus Linnaeus came up with was the genus and species system of naming. And so the genus is the first name, right? So like if you have a first and a last name, the first name would be the genus, and then the final name, so to speak, is the species. And they have to be written in a specific way. So let's take a common example. How about the human, right? Maybe some of you already have heard of the genus and species name of us humans, right? So the genus is Homo. So we'll write that down. Homo. Okay. Notice we capitalized that name, right? Just like you would capitalize your first name. And then we need to underline, all right? So if you name a genus, you should always capitalize the first letter and then underline it. Underline the whole word. And so you get Homo. Now, species for us is sapiens, Homo sapiens. But when we talk about species, or we write it down, we need to lowercase even the first letter in sapiens. And so by this convention, we would write it like this. Okay? And so we get Homo sapiens for us humans. And if in fact you're typing as opposed to, say, writing it by hand, so you're at your computer, then what you would do is you could follow this exact convention, or you could actually italicize. So you could hit Control-I and type this out without the underlining. One of the things, though, that you do is one or the other, or. We don't italicize and underline. So we wouldn't make this italics and have these underlinings here. That would be excessive. If you do something like that, the reader is going to see something's not quite right. Okay? Part of what you guys are here to do is learn at a college level of education, right? I mean, you're training to be professionals. We want to communicate professionally, and this shows that you know grammatically what you're doing. If you make mistakes on these basic conventions of naming, it's going to look strange or unprofessional. So we always follow these conventions, all right? Now, scientific names, they can communicate quite a bit about the organisms too. So Escherichia coli, right? Good old E. coli, as we hear about it, right? E. coli can be written like this, okay? But Escherichia, which is what the E stands for, refers to Escherich. Theodore Escherich was a microbiologist in the 1800s who is honored with, discovering this most studied organism on the planet. We know more about E. coli than any other organism, really, and that's kind of a uniquely a unique honor for him. And everybody has it. So 100% of humans carry E. coli in our gut. It's part of what it is to be human, is to have E. coli. The coli, by the way, then tells us where it lives, right? It lives in your colon. Now, if E. coli gets other places, you can be in big trouble, right? This can happen where, a person gets it in their blood and goes septic, so to speak. You can have it in your urine, and that's called, can cause a UTI. And so E. coli can get places where it definitely doesn't belong. But at least we know where it comes from just by looking at its name. Now, Staphylococcus aureus, this is a different genus and species, but you can look at the genus name Staphylococcus, and we can learn something from it. Staphylo is referring to, what does it say here? Cluster, grape-like clusters, staphylo. And then coccus refers to the shape of those clusters of cells. And so we have spherical cells there, and they're in bunches, grape-like clusters. And so we get Staphylococcus, and then aureus. And aureus refers to gold in color. Maybe some of you know the atomic symbol for gold. So if you take in chem class, or if you remember back to any chemistry, on the periodic table, Au stands for gold. And so when we look at these organisms grown on a plate, like on a petri dish, as we'll see during this course, they have this golden yellow pigment. And so aureus tells us about their color. And there are really some fascinating research topics that surround why these things look the way they do. Why do they have that golden pigment color? We'll talk about that later. And so these are things that we can know just by the name alone. And I wish I could tell you that scientific naming was going to give us ever more information, and that it was all perfectly standardized. But these things were named at different times during different centuries and eras and time periods. So the way in which they're named can tell us something, but not always the same things, classes of information that we need to know. Yeah. Oh, by the way, Staphylococcus takes me back. Just the name, because it's called staph for short, right? So we talk about staph infections. People can get these nasty staph infections. But when you look through the microscope at those, clinical samples where people have, say staph infection on their skin, they look like this, the grape-like clusters. And when I was a student, first time I took this course, my teacher's lab manual that she had written was called The Grapes of Staph, a real classic. So anyway, yeah. All right, moving forward. Just a little bit more about scientific naming and the conventions of it. Let's look through this sentence together here. After the first use, scientific names may be abbreviated with the first letter of the genus and then the species sort of spelled out, okay? Sometimes species is called specific epithet. That's just another name. I really just say species all semester, so. But let's look at this example. Escherichia coli. You have to spell it out fully the first time. And see, it's italicized, so we don't need to underline it, okay? And Staphylococcus aureus are found in the human body. Now, after we've established that in whatever communication, whether it's a research paper or you're writing an essay or something for your micro assignment or exam or something like that. You could say E. coli, E. coli, or S. aureus. And where are they found? So we already know coli refers to the colon, which is part of the intestines. So both of these would be good answers, right? Intestines or the colon, more specifically. And then Staph aureus is normally found on the skin. That doesn't mean we can't find staph elsewhere, right? So it is a skin pathogen, but it can get into people's blood and it can go septic. It can get into the urine and cause UTI, just like E. coli, but it comes from a different origin. And staph can get many other places in the body. So it's good to keep in mind where they generally are supposed to be, where they're naturally found, and then we know that they can cause serious infections. All right, now time to get into it. Types of microorganisms. There are so many. There are more than half a dozen, as we're going to see, and you're going to be responsible for knowing that list, and not only just knowing which types of microorganisms there are, but how they're defined. And so we're going to go through that. The first thing I'd like to do, though, before we actually define each microorganism, is talk about size. Okay. Here is the list, by the way. Okay, this will just give us an example of, a sort of order in which we're going to cover the microorganisms. Okay, but I wanted to compare their size. Okay, so when something's microscopic, like everything that's listed in this image, in real life, these things you would need a microscope to see them, but yet invisible isn't all created equal, right? So how small is small? Let's look at a yeast cell, okay? The yeast cell is the giant blue oval structure that we see here. And in fact, the yeast cell is very similar in size to a human red blood cell. So I'll go ahead and write that down here just for comparison's sake. Some people might like to think of a human red blood cell because RBC, I'll put RBC, red blood cell, or I'll put here human red blood cell, is almost the same size. Red blood cell is about 7.5 micrometers. So we have to learn a little bit about nomenclature here as well. The letter mu is a Greek letter. It's not an English alphabet letter. It looks like one because it's a letter U shape, but it has a tail on the front. Okay, that's called the mu. And this Greek, or the U with the tail on the front means it's a mu. And that is referring to millions. Okay, so 7.5 millionths of a meter. A meter is like a little over a yard long. Take a million little slices of that, and 7.5 of them will equal a red blood cell, or close to what a yeast cell would be. So maybe we can write that down somewhere. That the letter mu, the mu is also called a micron, and it is 1 millionth. Of a meter. Okay, so if a micron equals a millionth of a meter, and we're taking a class called Microbiology, then most everything that we study is going to be based on the millionths of a meter in size. So we're going to refer to many different microorganisms and objects and such on their micron level. Okay, so that's something to be familiar with and keep in mind throughout the semester. For instance, the basic, most ordinary bacteria that we see in and on the human body things like streptococcus that can cause things like strep throat or eat holes in your teeth and cause cavities. They are about 1 micrometer spherical in size. And when we compare that to, say, good old E. coli is slightly rod-shaped. It is considered a rod, but it is a short, fat little rod that's about 1 micrometer tall by about 2 micrometers long. So we can see the relative sizes of these organisms. And there are bacteria that are certainly smaller than that, but that's kind of unusual, like rickettsias, which we'll talk about later. These are really unusual bacteria. Well, subject for later, okay? Now, let's look at the ordinary bacteria when compared to even smaller life forms like the viruses. Here is a drawing of a pox virus. And we're not talking about chicken pox. I know when we think about pox viruses, we think of chicken pox. But a pox virus is a family of viruses that includes smallpox. Smallpox was a virus. I say it was because it's the only virus that has been eliminated from humanity. There are no active smallpox infections, which is just absolutely incredible if you think about it. Through vaccination programs, we were able across the globe to finally eliminate it. The last known smallpox infection was 1976, and I think it was an Ethiopian child in Africa. So, long time ago. It's gone. There's a great book called The Demon in the Freezer, and it details how smallpox is still found in a freezer, actually more than one freezer, deep in the CDC, the Centers for Disease Control, where in the United States we have smallpox stored. I'm not exactly sure why, right? We don't know if researchers are just thinking one day somehow we'd be glad to re-engineer smallpox or do something with it, hopefully not weaponize it. That's what the Demon in the Freezer subject is about. The USSR, Russia, also had, has this stored in their somewhere in government facilities, so smallpox is a fascinating virus that we're going to learn more about because of its historical importance, but also understanding what smallpox viruses are. The chickenpox, though, is actually a herpes virus, and these are some of the largest viruses out there in the world, and surprisingly, chickenpox being a herpes family virus, you know how it is, right? They're the gift that keeps on giving. I mean, you get herpes, you don't ever really get rid of herpes. Herpes goes into your nerves and it hides there. And chickenpox comes back, as we're going to learn, shingles. Think about it. If you've ever heard of shingles, it's chickenpox being reactivated in your nerves and causing fiery pain and nerve damage. And so we'll talk about how that works later. As we go on down this list, I don't necessarily need to outline every one of these, but one of the most famous, as we kind of look at some of these, the rabies virus, looks like a bullet. It's actually pretty amazing. This, ironically, really does actually, when it gets into your body, like say through the bite of a bat, or like a rabid dog, or a skunk, or some kind of animal that's carrying rabies, this will travel your nerve pathways, and like a bullet, it eventually makes its way to the brain. Scary disease. No one really survives rabies unless you get the vaccinations. And so, this subject of vaccinations will get talked about more during the semester. Unfortunately, it's a hotly debated topic when, in fact, I think debating whether or not vaccines are a good thing or a bad thing has really set us back. We worry about vaccination safety, but as we tackle the subject later, we don't want to set ourselves back for the wrong reasons. A lot of people come up with concerns, some of which are valid, many of which have stopped people from deciding that vaccines were, a greater reward towards covering yourself versus a risk of maybe dying or, losing someone to a bad disease or just becoming, suffering or maimed, you name it. So polio's back on the rise. It's unbelievable. And speaking of polio is actually one of the smallest viruses ever studied or ever discovered. Okay, so polio isn't much larger than a molecule of hemoglobin, the oxygen-carrying protein in your blood, right? That carries oxygen and carbon dioxide, exchanges those. Hemoglobin is so tiny. And now we're not even talking about microns. These are classified as 1,000 times smaller in their units. They're nanometers. NM is for nanometer. And so sometimes we go into the nanoscale. If you see the NM, so N, let's see if I can fit this here, N equals nano. And nano is not the millionth of a meter, but billionth, right? So 1,000 times smaller than a million is a billionth of a meter. And so anyhow, when we look at this scale, we can see viruses are very small. Everything in this picture, though, is microscopic. So we would refer to these, especially the viruses that are classified and measured on the nanoscale, as being what are called ultramicroscopic. If you took one of our regular microscopes, like we use here in everyday labs to look at bacteria, we wouldn't really be able to see these microorganisms that are in the virus category, because they're what are called ultramicroscopic. So how do you see an organism that is ultramicroscopic? Ultramicroscopic. If a microscope can't see them, an ordinary microscope, then we have to go to extraordinary types of microscopes, where an electron microscope is needed. So viruses are viewed with generally electron microscopes. Okay. All right, so anyhow, this gives you an idea of how small can be, like 1,000 times smaller than just microscopic, or we can talk about things that are just barely in the realm of microscopic. But blood cells, even, you still need a microscope to be able to see them. So these are all either microscopic organisms, or in the case of viruses, they're ultramicroscopic. All right, now, I'd like to go through, and one at a time, let's talk about the different microorganisms as with respect to what defines them. So we know bacteria is one of the most important groups of microorganisms. We spend most of our time in Introduction to Microbiology talking about, learning about studying bacteria, all right? And first and foremost, we need to know that bacteria are classified as bacteria because they lack a nucleus, all right? Deep in the center of most living cells that we think about have a little kernel, right? A little extra spherical structure inside that contains the DNA, and that's referred to as the nucleus. But a prokaryotic cell is one that has no such nucleus. Karyon refers to the kernel, and prokaryon, or prokaryote, is referring to before the kernel, before there was a nucleus. So the earliest cells that we know appeared on Earth literally billions of years ago were organisms that lacked the nucleus, and they still exist today. They're a successful life form. Bacteria are the most prolific, and they don't house their DNA in any sort of nuclear structure. Now, we should also keep in mind that differently than us humans, they also have a cell wall, right? So human cells have a nucleus. All human cells, pretty much, except for red blood cells, surprisingly. But we are nucleated creatures, cellularly speaking. But we lack cell walls, and And the bacteria lack a nucleus and they lack cell, but they do have cell walls. And what defines bacteria, for the most part, is their peptidoglycan in their cell wall. So in order for a bacterium to be a bacterium, it has to lack a nucleus. Generally, we'd say it has to have a cell wall, although there are some tiny exceptions, literally tiny exceptions, that we'll talk about. They're almost ultramicroscopic, they're so small. But besides those exceptions, bacteria have this chemical that makes up the cell wall called peptidoglycan. Now, for the sake of efficiency and just the fact that this is a kind of a tough word for students to learn about, you will have to get familiar with it. I think you should know the whole spelling and be able to spell it out, but I'll abbreviate it PG to make it a little easier to refer to sometimes. But you do need to memorize peptidoglycan since it's so unique to bacteria. All right, bacteria are unicellular by definition. Unicellular means single cell. And bacteria, the way that they make more of themselves through reproduction is by what's called binary fission. Binary fission means they just grow longer and pinch in half, and grow longer and pinch in half. It is a symmetrical sort of thing. And obviously this isn't how humans work. I mean, it'd be kind of crazy. Can you imagine, like, just grow longer and pinch in half? We're already like overpopulating, but that would be a whole other level of convenience. It's considered asexual, right? This is where you don't have to find a partner. All you have to do is grow bigger and pinch in half in equal halves. And this is what the bacteria do. And so we can see bacteria that are still connected. They don't always pinch clean in half, but they reproduce by growing longer and sort of dividing at a given place. They do have DNA. All complete living forms of microorganismal life has DNA, and that DNA for bacteria are big circles. Okay, they have one big loop of DNA in most cases. All right, and so that's good to keep in mind. Also, the way in which bacteria make their living can be defined in the widest variety of possibilities. All right, so some are photosynthetic. That means they, make their living by the sun's energy. Some of them then therefore would be like autotrophic, right? They're auto meaning self. Troph is referring to feeding. So an autotroph is one that doesn't need much to live, right? They're able to just absorb the sun's energy and then use some air and water, kind of like a plant, right? Photosynthetic, autotrophic, but others are heterotrophic, and we can talk about the different classes of heterotrophs, hetero meaning other, feeder, right? We can talk about the different classes of autotrophs even, so there are different ones, but the widest variety of life forms out there fall into the class of bacteria. Bacteria occupy the most diverse set of environments. I'll write that down. I had a professor, a bacterial physiology professor, who I'll never forget. One of the phrases he coined was, where there's a way, there's a bacterium. It's so true. Bacteria occupy the widest variety of environments on Earth. In fact, I would challenge you to think of a place where bacteria are definitely not present. So think about it. Can you think of one place where there's an environment where bacteria aren't found for sure? And I struggle to think of places where I can guarantee there are not bacteria. okay? Maybe an active lava flow. I don't know. I mean, that would be like one place that, yeah, okay, they're not going to be there. But there are very few places deep within the Earth's crust. Researchers and geologists have drilled down way below the surface of the Earth, and they've taken out core samples of layers of sediment, and through millions or billions of years, there are still bacteria that are found down there. Some of which are actually in a spore form. That means that they can be revived into an active living state, and so they're not always dead. It's really kind of incredible to see all the different places where bacteria can be found. You could take a jet way up into the uppermost reaches of the stratosphere, and you can find that bacteria are carried on wind currents. And that's something that we're still researching and finding more and more out about, which is that bacteria can be swept around, maybe due to storm activity or modern transport, and they're found even up in almost into outer space. So that's bacteria. Now let's compare bacteria to Archaea. All right, Archaea might make you think of the word archaic. If we say, "Wow, that's really archaic," right? That means like ancient. All right. I don't want to refer to someone as archaic, but you know what I mean. It's like an old idea, right? An archaic one. And yet, when we talk about this group of organisms, they are very much like bacteria. They're maybe almost as old as bacteria themselves. They are, evidence shows that maybe they've been around since, more than 1 billion years ago. Long time, billion with a B. So what makes them different than bacteria? In fact, speaking of archaic, back when I was a boy, right? Not that long ago in life. Okay, I'm not, I'm 44, so I would just say that when I was a kid going to school, they didn't talk about Archaea. They said Archaebacteria. But they were referring to this group that we now know is classified as actually separate. And so they are prokaryotic, just like bacteria. That's why we thought that they were the same as bacteria. But they're not, because they have cell walls, but the cell walls don't have peptidoglycan. So let's write that down. No PG in the CW, right? In the cell wall. So we'll put CW for cell wall, if that's okay. All right, and then what else? Well, maybe I'll denote that. If someone looks back at those notes, you might have a little issue there. So cell wall, right? I'll just put a little star there, and then we'll just put that there, like CW, so you know what we're talking about. Okay. Now, they are also unicellular, right? They're still single-celled. They reproduce by binary fission. They look just like bacteria. But the archaebacteria are primarily found in very specific environments, not necessarily quite as ubiquitous as normal bacteria. Ubiquitous, of course, means found everywhere, okay? We do find that most of the Archaea are classified as so-called extremophiles. Extreme in the sense that they can be found in places of high heat, right? Thermo refers to heat, so phile means lover. They're heat-loving, thermophilic types of Archaea. And there are some thermophilic bacteria as well, but a lot of them have been discovered or reclassified as Archaea. So, even up to beyond boiling temperatures. I don't know if you can even think of where is there a place on Earth in which temperatures could actually exceed boiling? And so you could think of deep at the bottom of the ocean, like on the sea floor where it's under pressure. And then there can be these deep sea thermal vents where, geothermal energy starts to cause the water to warm up down there, and it can spill certain nutrients and minerals out from the Earth's crust underneath the ocean, and these thermophiles can be found in places like that, or like in a geyser, like the one, I don't know, like Old Faithful or something, like, you know, go to Yellowstone National Park, and if you look at that big geyser, some of the water samples will show that thermophiles are found in there, and they're quite a fascinating group of microorganisms. Halophiles are also fascinating because these are involving places, environments where we figure that nothing could live. Like, what does halo mean? It means very high levels of salt. Like halophiles live in very high levels of salt. So salt can quite literally suck the water out of organisms. They dehydrate organisms when there's a lot of salt in the environment, and when salt is in a high level, we would think that nothing could live there. I'll give you an example. So the Dead Sea, maybe you guys, everybody's probably heard of the Dead Sea, or like Salt Lake in Salt Lake City, Utah, or we don't even have to go out of state. Here in California, there is, Mono Lake would be an example, all right? This is a lake that's so salty that, just like the Dead Sea, people just figured that there's nothing living in there, okay? But in fact, we now know that even though there are no fish because it's too salty, and there are no plants because it's too salty, when you take samples from these places, we can see that there are these microorganisms living in the water samples, and so that's pretty fascinating there. A third group of extremophiles, the Archaea, we don't have to go really anywhere to find. They're actually inside of us, okay? And the methanogens, which means, gen means to give birth to, and methano is referring to methane gas, right? Okay, so methanogens, they produce methane as a waste product, so natural gas, guys. Cows are a big source of methanogens, and in fact, this is a concern, a really big concern in today's environment especially. There are millions of cows across the globe, and probably the majority of them live in the United States with cattle ranch farming, and the problem is cows, if you've ever been out on a farm, I mean, how big is their waste, right? Cow patty, like huge amounts of waste, and they are filled with a lot of these methanogens. And cows' waste will give off methane gas. Methane gas is 4 times more powerful than CO2 at trapping greenhouse gases. I mean, trapping energy from the sun as a greenhouse gas. So methane is a problem for climate change. All right? Climate change influence. More powerful than CO2. 4 times more than CO2. I'm running out of room. Did this make sense with me here? Let's see if I can squeeze it in. Anyway, my point is, look, I'm not trying to talk you into being, vegetarian or vegan or something like that. It's just when we take inventory on different ways in which we can try to tackle the problem of too many greenhouse gases that trap the sun's energy and warm up our atmosphere and warm up the earth, we can at least realize that this is a driving force. It's something. I still like my In-N-Out burger, but we have to consider all the different things so that we can try to make a difference where we can, or how we decide we can. Different topic, I know, but it does involve microbiology, surprisingly. All right, now, fungi, the fungus among us, so to speak. Guys, fungi are very different than the first 2 groups that I've talked to you about so far, because they're eukaryotic, and that means they have a nucleus, all right? Remember, karyotic is referring to the kernel, right? The karyon, or the kernel. And EU means true. So like the true kernel, or having a nucleus at the center of each of these little fungal cells that we see pictured over here, whether they're molds or they're yeasts or even mushrooms. The kingdom fungi has their DNA protected in a nuclear structure. They also contain cell walls. So they're a little bit like bacteria and archaea in that regard. Humans don't have cell walls, keep in mind. But the cell wall that the fungi have is something called chitin. C-H-I-T-I-N. Chitin is also found in insect ectoskeletons. So like the hard outer shell of an insect is made of chitin. And if you think about it, that's a protective layer, right? That's what helps fungi to be really strong and kind of resistant to being destroyed in their environment. And you know how hard it is to kill mold, or even like yeast infections and such. I don't know if you've heard about molds or dealt with them in your lives, but fungi can be very difficult to get rid of. And at least in part, the chitin plays a role in their durability, their toughness. Now, they are considered heterotrophic by definition, all right? They do not use the sun's energy. No matter how much you might look at these things, especially like green mold, like penicillium or something, or like bread mold, or whatever that is growing there, they can have root-like structures. They can resemble plants a little bit, but they are not plants, especially because they do not use the sun's energy. All right, so I'm going to go ahead and write a term here in addition to in addition to heterotrophic, we're going to say non-photosynthetic to remind ourselves of how they're different than plants. All right, and they are sometimes unicellular. So think unicellular yeasts, kind of rhymes, unicellular yeast, or not rhymes, but starts with the same y, right? Alliteration, right? Okay? Or we can say multicellular molds. So unicellular yeasts, multicellular molds. We're not going to worry so much about the mushrooms, although they are multicellular. Microbiology doesn't deal with non-microscopic forms of life very often, so we'll just kind of leave mushrooms aside. All right. Anyhow, they are part of the kingdom fungi, so that's why they're there. Fungi have options. Fungi can reproduce sexually, but they have that ability when they can find a way to do so, but finding a partner for sex is obviously less convenient. Just you have to find somebody else, right? You have to find another one of your kind. Asexual reproduction, however, can happen much more often, I mean, think about it, right? You don't need to find anybody. You can just do this asexual thing, and in this case, it actually reproduces more of them. That's why there's so much mold around it. You know, allergies due to mold spores and yeasts and things that are hard to get rid of. They have ways of being proliferate, to proliferate that, you know, we humans don't have. They do have a similarity with us, though, when it comes to their genes. Their DNA is not circular like the bacteria and the archaea, but it is linear. So think about it, like females are XX chromosomes, right? The male has the XY. Those are all, you know, linear types of shapes and things, and fungi have this designation of how their DNA is shaped and packaged. All right. How about the protozoans? Now we're getting closer to forms of life that we're even more familiar with in terms of being human-like, okay? They are eukaryotic, just like we are, so they have nuclei. They usually lack cell walls. Now, protozoans are kind of an interesting bunch in general because proto means early, and zoa is referring to like zoo. Like, for instance, if you're a zoologist, then what do you study? Right? So like a zoologist studies animals, like a microbiologist studies microbes, a zoologist studies animals. Proto meaning early, so protozoan means like early animals. When life started on Earth, There were the bacteria that were the earliest life forms, but eventually protozoans developed way before humans. I mean, millions and millions of years ago, protozoans. In fact, even let's see, if we go back in our records, as we look through chapter 1, protozoans could be 1 billion years old in terms of how long ago they appeared. At any rate, they resemble the kinds of animal cells, but the difference is us as animals we're multicellular, whereas when we look at protozoans, they're unicellular. Okay, protozoans are kind of a mishmash, weird bunch because they're usually heterotrophic, meaning they don't make their own food, but there are some examples of little protozoans that can have chlorophyll in them. Right, and that's pretty amazing. Like Euglena would be an example if you've ever heard of those, like in high school biology or something. And they're strange because they're single-celled and they are a protozoa, but then they have the ability to use the sun's energy. But they also can eat, so they're autotrophs and heterotrophs. Bizarre. Anyway, so we won't really worry too much about that statement. But they're unicellular. They can reproduce sexually or asexually, so they have options, and they're found throughout the world. Some cause terrible disease. For instance, look at this one over here. That's an amoeba, all right. And an amoeba is amorphous. It doesn't have regular shape. It can change its shape and extend what's called a pseudopod, meaning pseudo meaning false and pod meaning foot, like so they can kind of extend like a so-called false foot, and they can pull themselves forward, and they can keep creeping along and pulling themselves forward. Or they can reach out more than one pseudopod perhaps, and then surround some food particles or something that they want to engulf and then eat. And so pseudopods are important for something called motility. Okay, motility is a technical word for the ability to move. All right, motility structures. I'm going to run out of room for that. Let me try that again. Motility structures. These are an important part of how scientists, taxonomists, biologists of different varying sorts have classified the protozoans is by how they move. Okay, so pseudopods is one. Flagella would be another. Okay, these right here are organisms that are that cause African sleeping sickness. All right, so these eel-like creatures can swim through the blood. These are a picture of some red blood cells. This is computer enhanced. It's not. I mean, it's realistic, but it's been colorized. But these organisms here are what are called hemoflagellates, and the trypanosome, as their genus is referred to, has this flagellum, and that single polar flagellum is how they whip their selves through the blood or whatever environment they're swimming through. Cilia, this is another example of a structure that is kind of like flagella, but they're numerous and they're found all over the body, and they beat in unison or in waves, and they allow organisms to move yet in that fashion as opposed to these previous two. Some protozoans, though, can be the deadliest of the bunch, really. I mean, there are bad guys in all these categories. There is a ciliated organism called Valentinium that can cause diarrhea and ruin your vacation. They're not found in the United States, that's why I say vacation. Dirty water in countries that struggle with poor sanitation. The flagellated organisms, African sleeping sickness, you have to go to Africa and actually get bitten by a black fly. Pseudopods, these guys, the amoeba, you can have that problem on vacation, but later we'll talk about the brain-eating amoeba that can go up your nose, in your nasal passage from fresh water, and that's happened right here in the United States. Deadly stuff. But the non-modal guys you can think of, they don't need any kind of way of moving. A mosquito or something like that can fly these organisms and then transfer them through a bite, and so you can get like malaria. And those organisms would be non-modal. The Plasmodium species, as they're called, or Plasmodium genus, is a non-modal example. You don't have to know the examples of these right now, but later on we'll talk more about them. So I'm just kind of trying to introduce these topics a bit. So that's how we classify protozoans, is how they move. Okay, now algae are yet another type of microorganism, all right? The algae are plant-like. So if protozoans may have been the earliest life form that eventually gave rise to animals, we would say that algae are the earliest life forms that gave rise to plants, okay? So algae, you can see, I mean, they have chlorophyll, they have pigments that match what we see in plants. And they are eukaryotic, so just like plants, they are higher life forms. They have nuclei that house their DNA and protect it. They have cell walls that contain cellulose, and so that's important for algae, just like plants have cellulose. They are photosynthetic, so by definition they have to use the sun's energy to make a living. They have to have access to light. They can be either unicellular or multicellular. They can even be a different variety of multicellular that's not always like you might think of multicellularity. I'll give you an example. So all of us humans are very much multicellular, right? I mean, it's kind of even hard to estimate how many cells make up the human body. We could try to look that up later. I mean, billions, you know, are there a trillion? Yeah, possibly. Trillion plus cells that make up the human body, but you can't just simply take one of our cells from our big multicellular makeup and like transplant it into some environment and grow a whole new you. However, with colonial algae, like this one here, okay, this is called Volvox. You don't have to memorize that right now, but I'll write it just for, in case you were wondering, well, what is this? Okay, this is called Volvox. Volvox is a colonial alga. It's colonial. And colonial is a type of multicellular. A type of multicellularity. Multicellular, but a single cell can cause reproduction. Or how about we just put can reproduce? Nah, I like can cause reproduction. So think how different that is from us humans, right? You know, you can't just pluck a cell out of the human body and just grow a whole new human. That doesn't naturally occur. We don't even have the biotechnology to do that yet, although who knows someday, right? And yet Volvox, you could take one of the single cells out of this shell that it's formed. They're differentiated cells. You could just pluck one of these cells out and transplant it into another environment, and if it had sunlight and water and enough nutrients or whatever, it could grow a whole new multicellular Volvox. That's an interesting point, but regardless of that, here's some algae down here. This is more like, you know, just look at this like, oh, that looks like seaweed, right? That's a type of green algae that it's not a plant, no matter how much it looks like a plant. This is a green alga. green algae here. And we can say that it is the green algae that are thought to have given rise to terrestrial plants. So they look like plants, even though they're not classified as plants, because they lack a true vascular system. So for instance, this does not have true roots. They can have root-like structures called holdfasts in the ocean that allow them to stay in one place to anchor themselves. But in fact, this floats around in the ocean without having traditional xylem and phloem, kind of analogous to like human blood vessels. These types of organisms just absorb through all parts of their structures without a true vascular system. So they do have the same chlorophyll though as terrestrial, or what I'm referring to as land plants. So green algae gave rise. We believe green algae gave rise to plants. Okay, and we can talk about algae as classifying them based on the color of the algae. So green algae is familiar because they look like plants and they give rise to plants. Red algae looks red, and we can talk about like red tide. Definitely we notice that when the ocean gets that reddish tint, and red tide can be problematic, right? Because some of these algae can carry toxins. And so if filter feeding fish, or like say shellfish, so like little crabs or shrimp or lobsters or something, right? Or oysters and mussels, you name it. If they are situated in red tide, and what that means is then there are these red algae called dinoflagellates. It's a little single-celled red alga that the shellfish can eat. And what happens is they concentrate the toxins. It's called saxitoxin. You don't need to know that, but it's a strangely named toxin, S-A-X-I toxin. Saxitoxin can be then act as a neurotoxin when someone eats like a plate of shrimp or mussels or clams or whatever. So red algae, are kind of an interesting group. By the way, red tide doesn't come around all year long. All right, red tide, they say you should not eat shellfish in the months that lack an R. It's kind of, I don't know if this isn't necessarily, I think, the best hard fast rule, but what would that be like? May, June, July, August. No R's, those are hotter months. They can bring about red tide. And in fact, we had some of the most amazing bioluminescence in red tide this year, 2020. And so that's kind of an interesting little bit about red algae. Brown algae are the big, usually like the big kelp that you see. When you see big seaweed or kelp out in the ocean, sometimes it's called ulva, U-L-V-A. That's the brown algae that we're most familiar with. So those are the way that we classify algae. And the colors come from the various pigments that are called accessory pigments in the chlorophyll that absorbs the sun's energy. Okay. Now, we're almost done, guys, with this part of the lecture. Viruses are a part of the groups of microorganisms, although they are what are called acellular. They are not complete life forms. Acellular means that they are not complete life forms. They depend upon other life forms, other living, actual living cells, to carry out their mission of reproduction. Okay. And so here's an example. I mean, look at this. This is an awful picture. This is a picture that generally can be thought of as a picture of death, in the sense that it's a T cell. All right, T cells are white blood cells found in the human immune system. They're found in the blood when a person gets infected with HIV, which we'll talk more about later. But like sexual activity, shooting up needles, or like through contaminated blood products or something, usually through injectable drug use like heroin, and shooting up, sharing needles, really big risk, as well as just sexual activity, unprotected sex. Sexual secretions can carry these little human immunodeficiency viruses, HIV, and all these little red dots that we see, I can't even count all of those. They're emerging from an infected T cell, and then these travel to, those little red viruses are going to travel then to other T cells, and pretty soon a person loses a part of their immune function that's vital to just fighting off ordinary organisms that wouldn't even be pathogens. It's really wild how bad viruses can be. Sometimes they're devastating. I mean, look at what coronavirus is doing to us. That's a whole other subject that I imagine at some point we'll spend extra time on, of course, because, well, that's why we're here on video right now. So we'll talk a lot about coronavirus when time permits. But these viruses, they have to have cells to infect. They are what are called obligate, intracellular, meaning inside cells, and they're parasites. The virus profits alone. Viruses do not bring any benefit to us humans, as far as we know at this point in time. So viruses are parasitic. They bring either DNA or RNA with them. All the medically important viruses are either DNA-based or RNA-based, but not both. Okay? There are some strange environmental examples that our book mentions that have been discovered more recently, where a couple of huge non-human pathogens have been found to have both DNA and RNA. You can look it up if you like. They're called mini-viruses, or the mama virus, which is like the mother of all viruses, based on the fact that they're a very large virus that contains both. But we're going to go ahead and just go with the DNA or RNA, but never both convention, because I try to focus this in on medically important viruses. All right, viruses that will infect humans and animals. All right, viruses also may be enveloped or naked. The envelope is like an extra outer covering, and so that covering is usually made of what are called lipid-based phospholipids. I'll put here the envelope is made of fragile lipids. Well, they're called phospholipids, but they're very easy to disrupt. Most viruses will die fairly easily with a disinfectant or an antiseptic. So the coronavirus would be an example of an enveloped virus. The HIV virus, the Zika virus, which is carried by a mosquito. All of these different viruses, the vast majority of human and animal viruses are enveloped, and they're covered in these so-called phospholipids that can actually be disrupted pretty readily. So that's a good thing, right? A little disinfectant should destroy these organisms before they get to you, if you have a chance to apply an antiseptic or disinfectant. At a minimum, though, all viruses, whether they're enveloped or naked, which means they don't have an envelope, they all have to have a capsid. All right, the capsid is a protein shell or a protein coating that contains the DNA or the RNA and protects the virus. At least protects its genetic code. All right, so that's as much as I want to say about viruses for now. We're going to talk a lot more about viruses during the virology section, and when topics about coronavirus come up, because with the pandemic going on. All right, and last but not least, guys, the last group of microorganisms to end our first part of this video for the first day is to talk about the multicellular animal parasites, okay? Helminths, worms, that's just a technical word for worms, come in a variety of shapes and sizes. Some worms are gigantic. This is a tapeworm. If a tapeworm comes out of an animal or an infected person, this could be like 25 feet long. I mean, that's unreal to think about, but yeah, some worms can be so long that you don't need a microscope to see them. You would only need a microscope to see their eggs and some of their internal parts. Of course, this heartworm, which infected, this is a horse's heart, it's an animal heart, and you can see the heartworm that infected this animal. But whatever the case, worms can be flat, all right? They can be round, like an earthworm, okay? And we'll talk about their specific names later. But they are animals, and they're multicellular, not unicellular, and they have no cell wall because they're animals, okay? They do not use the sun's energy. These things are living in the intestines and the heart and the organs. I mean, they're anywhere but not where the sun is shining, right? So they definitely are not depending on the sun. They can reproduce sexually or asexually, which is a tough part of their life cycle. We'll talk more about that as we learn about the way that they reproduce. And speaking of the way they reproduce, it's the microscopic stages, the larvae and the eggs that concern us in microbiology. And that's why this is a topic in microbiology, is because of the larvae and the eggs. All right, those are microscopic, and so we need to know about how they reproduce and how these organisms infect us invisibly through the microscopic stages, and so they're a part of microbiology, and we'll learn about them later in the semester. This is an exciting topic. It's part of what's called parasitology, which is separate from talking about bacteria and viruses and such, and so and even fungi. So this is a very separate topic that we'll get to the last part of the semester. Okay, so I think we're going to stop here. This is a sort of just a little quiz question, and I was thinking that maybe we can go over some of this during our Zoom conference. And so it asks which of the following is not a characteristic of bacteria, right? And so you can kind of go through the things that we've learned so far, and so what would you choose, right? So we'll look back at this, okay? There's also a table here, and this table says comparison of organisms, all right? This will be filled in so that, well, I would like you to spend some time trying to fill this in, and then we can verify your answers, right? So during discussion, we can look at this, and you can see whether or not you got the right answers to all the different designations of or characteristics of the different groups of microorganisms and how they fall into this category. And what comes next is something about classification, but I'm going to start up on that next time and press pause until later, or until next time.